In this screencast, we are going to talk about some important mono, di, and polysaccharides, and then talk about some oligosaccharides at the very end that are important. We're going to start with monosaccharides. There are two pentoses, ribose, which is shown here, and deoxyribose, which is shown here, that are important because they're used in the synthesis of nucleic acids. So we find these in nucleic acids, which are DNA and RNA, right? And they're also, this DNA and RNA are really essential in protein synthesis and the transfer of genetic materials. And we're going to talk about that again in chapter 21. Ribose, along with a phosphate group, forms the long chains that make up ribonucleic acid. So this is what we find in RNA. And then deoxyribose, along with the phosphate groups, which is shown here, this is what I have to show for you here with this, forms DNA. When we look at this particular image here, the S's are those sugars, and then we have phosphates that link them. And then the nucleotides, the base pairs, are right in here in the center. And so this is what makes up DNA. But as far as our important monosaccharides are concerned, it's these pentoses that are important. So ribose and deoxyribose are pentoses. And one of the things I want you to notice is when we talk about deoxyribose, it's missing the OH in the 2 position, right? So that's how it differs from ribose. So I'm going to say in the 2 position, but you could also say the second carbon. The next important sugar that we're going to talk about is glucose. Of the monosaccharides, glucose is the most important nutritionally and the most abundant in nature. So this is important nutritionally and the most abundant in nature. We find glucose in things like honey. It's found in fruits like grapes, figs, dates, any sort of fruits like that. Ripe grapes, like what you see here, are about 20 to 30% glucose. This is also called sometimes dextrose. And it's also known as blood sugar. So if you're testing your blood sugar, you're testing the amount of glucose in it. This is the final form where all other sugars absorbed in the body are converted to in the liver. So other sugars absorbed into the body must be converted to glucose in the liver. Glucose is commonly used as a sweetener in a lot of our foods. When we look at glucose, the form that we're looking at is beta-D-glucose here. This is the open chain form, and this is the form with the ring here. So glucose is a very important monosaccharide. Another monosaccharide that we find is galactose. Galactose is has a structure that's very similar to glucose. The only difference between the two is the orientation of the hydroxy group attached to carbon 4 here. The OH here is down in glucose, and for galactose, that OH is up. When we look at this is the open chain form of galactose, and this is the ring form of galactose. So This is synthesized in the mammary glands where it's incorporated into lactose, the sugar found in milk. It's also a component of substances present in nerve tissue. So this is a component of lactose, which is a disaccharide, which is milk sugar. And it's synthesized in the mammary glands. And it's a component that's also present in nerve tissue. The next monosaccharide that we're going to talk about is fructose. Fructose is really the most important ketohexose that we're going to discuss. It's also known as levulose, right? And it's also known as fruit sugar because it's of its presence in lots and lots of fruits. It's present in honey, which is shown kind of right here with some fruit, in a ratio with glucose, and is abundant in corn syrup. This is the sweetest of the common sugars. And is important as a food sweetener because less fructose is needed than other sugars to achieve the same degree of sweetness. So I'm making sure we've got kind of all of that down. It's a ketohexose. It's present in honey in a 1-to-1 ratio with glucose, and it's also abundant in corn syrup. Now, let's talk about some disaccharides. When we talk about disaccharides, these are just two monosaccharide units linked together by either an acetal or a ketal, which remember we're going to call these glycosidic linkages. Now, a glycosidic linkage, I just want to remind you, is identified by, right, the number that is associated with the carbon atoms joining together the linkage. So the configuration for any linkage is also given. So for example, this is going down, and so that's an alpha, and we're connecting the 1 carbon to the 4 carbon here. So this is an alpha 1,4 linkage. I also want you to notice when we look at this, we have a cyclic hemiacetal here. Another name for this is a free anomeric carbon. And if you have a free anomeric carbon, then you are a reducing sugar. You'll react with Benedict's reagent. Now, these can be hydrolyzed. So this is maltose, for example. It's one of our diet saccharides. It can be hydrolyzed back to yield their monosaccharides by boiling with dilute acid or reacting them with enzymes. And a lot of times our body breaks these down using enzymes. Nutritionally, the most important members of this group are maltose, lactose, and sucrose, and those are the ones that we're going to look at. So we're going to look at maltose first. Maltose is also known as malt sugar. It contains 2 D-glucose units joined by an alpha 1,4 glycosidic linkage. Now, I want to tell you something about how that 1,4 is written. When we write it, I've also seen it written as alpha 1, 4 glycosidic linkage. So you can use either one. You need to be able to recognize both. Your book tends to use that top one. Okay. This maltose is found in germinating grain. And is also formed during the digestion of starch to glucose. That is a reducing sugar because we have a free anomeric carbon. Right? And it will form 2 units of D-glucose upon hydrolysis. So if this goes through a reaction where these bonds are broken, it's hydrolyzed, we will get 2 D-glucose units out of that. I'm trying to think if there's anything else I want to tell you about maltose. Well, let's go on to lactose. Lactose is also known as milk sugar. If you are lactose intolerant, what that means is you lack the enzyme to break this bond here. All right. And so this is found in, it's about 5% of cow milk and 7% of human milk by weight. It's composed of galactose and glucose units joined by a beta 1,4 glycosidic linkage. It's a reducing sugar because it has that free anomeric carbon. All right, so, here is our free anomeric carbon, and so that's why it's considered to be a reducing sugar. And of course, this is found in foods like milk, cheese, those types of things. Let's talk about sucrose. Sucrose is commonly known as household sugar. It's composed of fructose and glucose units joined by an alpha 1 beta 2 glycosidic linkage. Okay. It's found in the juices of plants, especially sugarcane and beet sugar. So in beets. If you go into the grocery store and you buy sugar, you're going to see some that are labeled as cane sugar, and this comes from sugar cane. But you're going to see some that are not labeled, and if they're not labeled, they are coming from beets or beet sugar. It is not a reducing sugar. It does not have a free anomeric carbon. And what I mean by that is both the anomeric carbon here and here are involved in that glycosidic bond. So they will not react with Benedict's solution. So you can see here, I've drawn this as an alpha 1 beta 2 glycosidic bond with an arrow. You can also see sometimes this is written as alpha comma beta 1, 2 like that, or sometimes I've seen it written as alpha comma beta 1 comma 2 like that. So there's a couple different ways it can be written, which is what makes this a little tricky. This is hydrolyzed to produce invert sugar. Invert sugar is just a mixture of equal amounts of glucose and fructose. Now, just as two sugar units can be linked together to form a disaccharide, we can link additional units to form larger saccharides like oligosaccharides or polysaccharides. When we do this, these are condensation polymers containing thousands of units to form a polysaccharide. Because their size, polysaccharides are not water soluble, but their many hydroxy groups become hydrated individually when they are exposed to water. And so some polysaccharides will form thick colloidal dispersions with heated in water. So if we think about this, a really common polysaccharide is starch, which we found is in potatoes. It can also be used as a thickener in sauces and gravies and pie fillings and any sort of food preparation. One of the things that I find really interesting are the differences in the properties of monosaccharides and polysaccharides. The molecular weight of monosaccharides and polysaccharides are relatively low compared to polysaccharides, which are very high. Monosaccharides and disaccharides are very sweet. Your book says polysaccharides are kind of tasteless, which is why we can use starch, for example, as a thickener because it doesn't affect the taste. These monosaccharides and disaccharides are soluble in water. Polysaccharides are insoluble or form these colloidal dispersions or kind of like these thick pastes. The size of particles here, the monosaccharides and disaccharides will pass through a membrane. Polysaccharides will not pass through a membrane, and that becomes important in the next few slides when we talk about some starch tests. And then if we're using Benedict's solution to test for reducing sugars, monosaccharides and disaccharides are positive except for sucrose, but polysaccharides are going to be negative because all of their anomeric carbons are used up. So I want to give you some kind of just information about polysaccharides here. So when we talk about polysaccharides, these are just condensation polymers that contain thousands of units. They are not water soluble, as we mentioned, but they become hydrated individually when exposed to water, which we talk about right here. And so they can form those thick colloidal dispersions. A really good example is starch. And so because of these properties, it can be used as a thickener, thickener in sauces, gravies, pie fillings, and other food preparations because it's sort of tasteless and so it doesn't affect the taste. So let's kind of zoom in and talk about starch a little bit. So when we talk about starch, this is a polymer that consists entirely of D-glucose units. So it's a polymer that consists entirely of D-glucose units. It's the major storage form of D-glucose in plants. And it has kind of two starch fractions, amylose and amylopectin. So it's got two components, right? One is amylose. Okay. And when we talk about amylose here, right, that's made up of long unbranched chains of glucose connected by alpha-1,4 glycosidic linkages. So amylose is an unbranched chain of 1,000 to 2,000 glucose units with alpha 1,4 glycosidic linkages. It makes up about 10 to 20% of starch. Then the next kind of component of starch is amylopectin. And when we look and we talk about amylopectin, it's a branch chain. So whereas amylose is linear, now we're branched. And that consists of alpha-1,4 linkages, but it also has those alpha-1,6 linkages, that glycosidic linkages that cause the branching. And it makes up about 80 to 90% starch. So when we look at the structure here, we can see, so we have a potato, which is where we have starch, and starch is made up of about 10 to 20% amylose and 80 to 90% amylopectin. These give really specific percentages, but we know there's a range, and it's made up, each of these is made up of those D-glucose monomers. The amylose is the unbranched chain here. It's a straight chain. And then the amylopectin has those alpha-1,4 glycosidic bonds, but every so often it's got an alpha-1,6, which then ends up with these branches. And so even though it's linear, it kind of coils up here, the amylose does. And then we see kind of where the amylopectin also curls, and then there's these pieces that are sticking off from it there. So I want to show you a slide kind of with larger structures, amylose and amylopectin, so that you can see it just really well, and particularly see how your book represents it. When we look here, sometimes these bonds are kind of represented as U's. I don't love that, but this is the way that your book does it, so I want you to be able to recognize that as well. These are the alpha-1,4 linkages, and this is the alpha-1,6 linkage. So that right there is that alpha-1,6 linkage, and all of these here are the alpha-1,4 linkages. And so that's the kind of the difference there. But there's amylose and amylopectin. Now, we can test for starch, and we test for starch using a reaction between iodine and that coiled form of amylose, which is what I pointed out on the previous slide. So the test for starch is just a reaction that occurs between iodine and the coiled form of amylose, right? And so when we look at this particular reaction, the product of it is a deep blue color. And is that deep blue color is because what happens is the amylose helix, which is that coiled form here, we just call this a helix, is filled with those iodine molecules. And so those iodine molecules are trapped and won't wash away. And so that results in that darkish blue color. Now, this reaction in particular is used to monitor the hydrolysis of starch. Because what happens is as the starch is hydrolyzed by either acid or enzymes to form smaller polysaccharides and maltose and then finally glucose, that color will fade because it's no longer being held here in the helix. So the color fades as the polysaccharide is hydrolyzed and broken down into smaller polysaccharides and then maltose and then eventually glucose. Right? And so the disappearance of that blue iodine color is because this starch helix is breaking down. Now, let's talk about another important polysaccharide, which is glycogen. This is sometimes called animal starch because it's a storage carbohydrate for animals that's analogous to the starch of plants. So it's also known as animal starch because it is a storage carbohydrate for animals that is analogous to the starch of plants, right? It's especially abundant in the liver, and the muscle, right? Where we have excess glucose taken in by the animal and stored for future use. So this is where excess glucose is taken in by an animal and stored for future use, right? On hydrolysis, glycogen forms D-glucose, which helps maintain the normal blood sugar level and provides the muscles with energy. So when it's hydrolyzed, right, it forms D-glucose, which helps maintain the normal blood sugar level and provides the muscles with energy. Now, structurally, glycogen is very similar to amylopectin. Right? Because it contains both those alpha 1, 4 and alpha 1, 6 linkages. Now, where it's different though is that these are going to be between 8 to 12 D-glucose units. Whereas amylopectin has the branching between 20 to 24 glucose units. And so that big difference then is that glycogen is more branched than amylopectin. And so that's the big difference between the two. Now, another important polysaccharide is cellulose. Cellulose is the most important structural polysaccharide and is the most abundant organic compound on Earth. So, it is a polymer of D-glucose. It's the most important structural polysaccharide and is the most abundant organic compound on Earth. All right. And the reason is it's the material that's in plant cell walls that provides that strength and rigidity. It has a linear polymer like amylose, but instead of alpha 1, 4 glycosidic linkages, it has beta 1, 4 glycosidic linkages. Right? But it's still, when we look here, is still linear, right? And then to get the structure, we see that the cellulose here will align in this rigid form to give the structure here, which we see, for example, in our fibers. So from like cotton and trees and things of that nature. Now, the beta here, the 1,4 glycosidic linkage that's beta, means that we cannot digest this. We don't have the enzyme that digests this. Right? And what it also means is that animals that do use cellulose as food only do so with the help of a bacteria that has the enzymes for breakdown. So animals that digest cellulose have a gut bacteria that can digest the beta 1,4 glycosidic linkages. Right? So herbivores like cows, sheep, and horses are in this category. Right? And each has a colony of this bacteria somewhere in their digestive system that uses the simple carbohydrates from that bacterial hydrolysis of cellulose. The soil also contains organisms with these enzymes. Otherwise, debris from dead plants would accumulate rather than decomposing in the forest through biodegradation. This really small difference causes just tremendous differences between amylose and cellulose. The shape of the molecules are really different, as I kind of pointed out here. Those alpha 1,4 linked amylose tend to form those really loose spiral structures, so they're very flexible. However, these beta linkages tend to form these extended straight chains, and they become aligned so well that they can form fibers that are really rigid and strong. Now, one thing that because we do not digest cellulose, one thing to be aware of is this is a constituent of dietary fiber that aids in intestine contraction. So because we cannot digest this, it is a part that dietary fiber is part of our diet because it's helpful with that intestine or intestinal contraction. So while we don't get any nutritional value from cellulose, it can still help us to aid the passage of food through the digestive system. The last polysaccharide we're going to talk about is chitin. Chitin makes up the exoskeleton of insects and crustaceans, as well as the cell wall of most fungi. It also has beta linkages, but these are, it also has beta 1,4 linkages. But it has a modification, and it's got a C2 modification here. This says it's an amine group that's attached, but we know that that's an amide, so I do want you to kind of fix that there. Okay, so we have this particular attachment here that's modified in all of the glucose units. And I want you to notice that when we look at a long chain here, it flips. So we'll have one on this side and then one on this side and so on and so forth. But that polysaccharide is used for the exoskeleton of these organisms as well as the cell wall of fungi. And then the last thing I wanted to talk about are red blood cells. Red blood cells have several different chemical markers, which are shown here, bonded to the cell, and these are called the ABO blood markers, and they contain 3 or 4 monosaccharides, and those monosaccharide structures are here. And so when we look here, depending on the blood type, here's our red blood cell, and on the surface of the red blood cell, if you're type O, you have N-acetylglucosamine, you have galactose, and you have fucose. If you're type A, you have those same 3, but off of your galactose is another N-acetylglucosamine group, but it's N-acetylgalactosamine. And then if you have type B, you have a second galactose molecule here. Now, all the blood types include carbohydrates that N-acetylglucosamine, galactose, and fucose. Now, type O contains only those 3 carbohydrates that are attached to the surface of the red blood cell through that glycosidic bond. And type A and B contain those plus that 4th carbohydrate. So what this means, right, is that type O is considered to be a universal donor because these are the same 3 here, here, and here. So you can donate type O blood to type O, to type A, to type B, or to type AB, which is a combination of these 2. Okay, and so it's a universal donor. It can be donated to any of those. Now, that type AB is considered to be a universal acceptor because it can accept blood, or if you're type AB, you can accept blood from someone who's type O, type A, or type B, or type AB. But someone who's type O cannot accept blood from somebody who's type A because of this additional unit here. The body doesn't recognize that and will reject that blood transfer. And so these are examples of some of our oligosaccharides, which are really important in particularly blood typing. So this ends for us our discussion of kind of important monosaccharides, disaccharides, polysaccharides, and oligosaccharides. And it also ends our discussion of the carbohydrate chapter. As always, if you have any questions, please feel free to reach out and stop by my office.